Spring entertainment. They got, they're always selling knives down there. I don't know. I might drive through there. Huh?
We're staying at a friend's parents.
Yeah, it's a pretty area, so that should be nice. Anyone else doing stuff? Anyone going to Hawaii? I hope not. We were in Mexico a few weeks ago and a hurricane missed us by 300 miles. I don't know if Hawaii is going to be as lucky, but so book that for Christmas. You had something?
I'm going to Vegas.
Vegas. All right. Give him money. Gamble for us. Help the state budget. So, well, that's pretty cool. Anyone? I'm hanging out. My wife's leaving me, but she'll be back. She's going to Oklahoma to visit her family, so that's real cool. But I kind of got stuff to do here, so that all worked out okay. Okay, well, good on that. Nice morning. Let's get some science done, and then we can go out and do other things, whatever those might be. So where are we at? We're starting with the cell. The prokaryotic cell is our main interest, although we're kind of, every once in a while looking at the eukaryotes, the microbiology. The way it is, we have to keep our eye on the eukaryotes, but our emphasis is in the prokaryotic world. And we start thinking about the cell membrane, and we think about its importance as a permeable barrier, and how it organizes the cell, and how it even sets itself up to make the cell into a small battery, which is way cool. And we started thinking a little bit more about the cell membrane, and we said, well, the cell membrane is made out of a lipid bilayer. And if we take a little closer look, the bilayer is made out of what molecules? Phospholipids. So right away, we'll say, well, you need phosphorus to grow things. So that's at least one place we know it has to go besides to the nucleic acids. And we said, well, the phospholipids are pretty cool because they have they're hydrophobic and hydrophilic, and that's a big deal because they set up a type of stability just by their very simple chemical nature. I mean, and carbons and hydrogens and oxygens are pretty easy to get in the cell. They're not very complex, like we know with proteins and all those kinds of things. So that was pretty neat. And then we said, well, we know the cell membranes, the bacteria are like us. They have two fatty acids and some kind of phosphate moiety on that, and that has a polar disposition. And we know the fatty acids are linked to the linking molecule glycerol with what kind of linkage? Or maybe we don't know, but we're thinking now, what kind of bond or grouping is putting the fatty acid onto that trialcohol molecule we call glycerol? Sounds like ether. Ester, okay. And ester, okay. Ester's on there, and we lose a molecule of water, dehydration synthesis. That's old news for us. We learned that years ago. So that's cool. And that is a characteristic that we can look at in not only our cells, but in the bacteria. And so now these archaea, they're kind of neat, but they do things a little different. Rather than just having simple saturated and unsaturated fatty acids, they have a repeating group made out of 5 carbons called the isoprene that comes together and makes a fentanyl group. But that fentanyl group connects to the glycerol molecule with what linkage? And here's your chance. Ether linkage. So look that up. We basically have lost a double bonded O is the deal on that. You're like, why? Who knows? Well, usually things are selected for. It's probably for stability. And we know with the archaea, they said, well, they said as a group, they all got together and said, we either going to have a kind of a fentanyl bilayer, only with this ether group, or some of them go straight across, so we get a tetraether type of situation with that. So that's kind of where we left things. I think we got up to talking about gram-negatives and gram-positives, eh? Okay, so we've been through the general morphology, the general setup of the cell. And this is where we left it. It kind of looks like layer cake, 3 days left over. But we're going to go into things in a little bit more detail. So we've been drawing our attention now back to the bacteria, and then we've been through the lipid bilayer, and now we're going to start going through the other layers, all right? That'll probably take up a good part of the lecture today. We were, in review, talked about then first the gram-positive, and in my mind, probably the more simple of the morphological parts or types of the cells, because we do have, in review, the cell membrane. We realize this artist drew a blue line, but that has all the detail we talked about. It's got integrative proteins, some that go through, some that are within. It's a lipid bilayer. All those things are still in play. But then what's really interesting to us is this peptide sugar layer, okay, peptidoglycan. And then I think we got to the next slide and called her quits on Tuesday. And this is a nicer depiction of this. Very simple, but tells me a lot of information, and I like it. Okay, we've been through this. This is the inside of the cell then. As we move to the outside of the cell, there's kind of a little layer, and that's not a mistake. These aren't, even though there's connectors through this cell wall and the cell membrane, it's not like it's right on top of it. This space becomes really important, more important in the gram-negatives, and we'll visit those guys next, okay. But we're going to draw our attention to this layer, okay. This layer we see has repeating units. Now, this shows it generally. We're going to find out that this unit has a little squiggle in here, and this unit are not exactly the same. It's actually a disaccharide, a repeating disaccharide derivative. Okay, that will be coming up in a second. But what is also important is if you're to look at the different layers, they're anchored together. So we'll also find that this same kind of anchors the disaccharide derivatives together. So that's the kind of idea. If you could think of, for a moment, instead of looking in a cross-section, but looking down and imagining like a t-shirt or a fabric, I don't care, but pick your favorite fabric. You know there's strands that run one way and strands that run, let's say, vertically and horizontally. Okay, we're holding it up like this. If you can imagine some of the strands being the sugar strands going through the fabric, okay, and then the other strands being peptides holding those strands together. They don't really weave as much as just kind of bond them and hold them in place. You get this great kind of matrix that is a lot like, I think, as a fabric. So if we have one layer of the fabric now going around the cell, this is another layer. And if you just break apart the matrix and thinking sugar and connecting peptides, you got peptidoglycan, and you got its function, and you start to get the economy of nature, just taking a glucose molecule and deriving it into that. So let's take a closer look. Where we're going now, just so you don't lose context, is we're going to start looking at these guys. Okay, now we're going to go magnify things about 12 billion times and look at it in the molecular level. We have this repeating, don't worry about this yet. We're going to put our focus right here. We have these repeating disaccharides that were like little hexagons in that last picture. You see some things you recognize. Oh yeah, that kind of looks like glucose. Oh, there's a beta-1,4 linkage. That's pretty interesting. Beta linkages are what we find in what glucose polymer? The most common polymer on Earth. Natural polymer, excuse me. Polyester might rival it at this point. But glycogen is a good first thought. Starch is alpha. Cellulose. Good. And we still see beta and chitin and those kinds of things. But yeah, those polymers are around. And so this 1,4 linkage is between the two glucose moieties. So, we think, well, we know in respiration we rip down glucose, but we also know glucose is around. It's one of those things that the cell, gets a lot of, to see it either manufactured or altered, isn't like a whole big type of pathway that the cell has to invent. So it's an adaptation from glucose, which is nice. And then the first part, the N-acetyl group, okay, N, not hard, acetate group on that is on carbon 2. That's simple, so that's N-acetylglucosamine. If you also just want to write a shorthand of NAG, N-A-G, I'm cool with that. If spelling is a problem for you as it is with me. Okay, the other one is N-acetylmuramic acid. They are putting a G and an M. I don't like using the G a lot, because sometimes students mix it up with guanine, so I usually use NAG and NAM. Okay, if you want to go that way, that's fine with me. I just want to make sure we know what we're talking about. Anyway, look at this part. Same. There's the N-acetyl group. Okay, not bad. You'll see the same kind of adaptation in chitin. You see a little bit of a nitrogen moiety on there, and it takes that kind of polymer, and we wrap it around almost every insect in the world. It's a really neat way of making structure. Okay, and then here's the other part. Muramic acid stops actually right here on this end of this carboxylic acid. Okay, this slide kind of moves ahead on this a little bit, but when we have that acid there, this part here then is muramic acid, and that's it. But what's neat about that, I can tell you a little story that might help you remember it. Here they have this enzyme called lysozyme, as it sounds, lysis things. Their name for lysozyme is muramidase. So a muramidase comes in and can break this bond. And you go, well, so what? Well, I mean, if this bond is holding together the fabric of the cell wall, that cell tends to fall apart, especially if it's growing and trying to manufacture it and can't link them together. You know where you have a good amount of muramidase as a natural bactericide? Several places in your body. Not too much your gut, so let's stay in other places. You make me cry. Tears. Tears have a lot of lysozyme or muramidase in them. In fact, we used to do this in lab. I don't know if the students got bored with it. We stopped doing it so much, but we would get a tear. I'd either tell one of my stupid jokes, or we'd get some onions and stuff, but we'd get someone to give us a tear, and we would dilute the tear out 1 to 10, 1 to 100, and so forth. And the lysozyme, and then we would plate it with some gram positives, and the lysozyme would be effective even up to 1/10,000th of the concentration in your eye. Now, it's not enough enzyme to hurt your eye, but it's just one of those things that, it's probably a good idea that we don't have big bacteria growing all the way through our eyes. That would kind of make driving a little bit more challenging than it already is. So there's one linkage I'd like to know about is not only the linkage between the carbohydrate or the saccharides, but then what breaks it. Okay, that's important. You can use that. Your body uses it. You have some in your saliva also. Not to as great a degree though. It's much smaller. But a lot of the way your face is put together is there's some overlapping mechanisms. Okay, so now we got that backbone. So if we go back, that backbone, and I got a few pictures on these things, but that backbone is this. So here's the muramic acid. We know that peptide thing is coming there. Here is the NAG, the acetylglucosamine. Okay, now off of this muramic acid, we have a peptide moiety. I could sit down and make you learn all this, but it wouldn't help you. That they tend to be different among species, except that they do have the peptide moiety, but they change in number, they change in composition. Sometimes we even see, like the diamino-pamelic acid is unique. It's one of those. It's not one of the main 20 amino acids that we usually learn about in someplace in biology. We see alanine. We see some of these other ones. We recognize that. But so this is going to make a bridge to the other sugar fiber, and then it's also, so it's making a bridge, so if it's sugar here and sugar there, it's going to make a bridge across that. It's also going to make a bridge to the next layer above it if there is one. Okay, so and I think that's where we're going. Yeah, so we have the glycan backbone. This is kind of for your notes. Try to animate it a little bit, and then the peptide linkage. I think we did that license. All right, talked about that. This is variable. Here is NAG and NAB, more creatively written, but we get the idea of this branching peptide, that when you put the peptides together, when you're forming the cell wall, the peptidoglycan, it's called transpeptidation, but basically putting a peptide bridge across things. Now here we go with, what is this? This is in coli, this is in staph, so you can see already variation, which is my point across there. But this diagram is worthwhile knowing. The G's and the M's are NAG and NAM again, but between the muramic acid, we're just looking at a single layer here, and we see those cross bridges. So this is fairly clever. I mean, if you like that economy of nature and making things, because really there's not a lot of variability here. And you have peptides and a simple carbohydrate readily available. You add a little nitrogen in there, that's fine. But you can really make a lot of this fairly fast as a cell, which is important because you're going through reproduction sometimes every 20 minutes, which is fairly remarkable. Your cells probably haven't done that since the time you were a little blastula or something. You're making all these different cells, and you look like a chicken, you look like a lizard, and then you look like you. So here we go, a little bit more color, but we get the idea. There's different ways of showing it. Here, nice and colorful, different disaccharides. This shows nicely those bridges between the NAM each time. Okay, so they are a little bit different for a very specific reason. Now, we'll come back. No, that's right, penicillin. So we talked about this bond between the sugars being affected by lysozyme, the transpeptidation process. That's where penicillin reacts. So antibiotics do different mechanisms. Some stop protein synthesis, okay, like tetracycline, oxacillin, some of those, and then some affect the way the ribosome works in the cell. Some affect ribosomes that are the size of bacteria, but not your ribosomes. That's why we like antibiotics, because they tend to act on the prokaryotes and not us. That's the problem when you get the eukaryotic disease. The cells are a lot like you, so you're partially killing them without killing you. But with penicillin, it's nice. It doesn't deal with the protein synthesis at all. It deals with the cell wall. So, and now if you're allergic to penicillin, they have other things. But that was the big breakthrough, is to find something that affects the cell wall. And we'll find in a little while that if you have more peptidoglycan, the penicillin's more effective. Penicillin won't work. It won't work on cells that are established, but when the cells are growing, they can't weave together, and so they explode. So it's cool and sad for the bacteria. Well, that's kind of it for the cell wall of the gram-positives. We know this part here. We talk about the cytoplasm. We know there's kind of a little space. We're going to call it a matrix. In the old literature, they thought it was a space because they were dehydrating everything and looking at it with EM, electron microscopy. We now know that there's a very slight matrix here. And here's all our layers, 1, 2, 3. There's a dozen or more layers of peptidoglycan that we talked about. That's all expertly woven together for each fabric line, as I call it, and then between each layer of the fabric. So that's pretty cool. We keep coming up against these inclusions. Teichoic acid, which has a fairly big ribitol component, and phosphate. I don't really, at this level, I don't think there's really any advantage for us to have to really look at the molecular structure except if you're interested, as scientists, you always want to know more, but I just like to say that it imparts a negative charge to the cell. Okay? Most bacteria are negatively charged, and you wonder about that. Well, if we want to get rid of bacteria, why don't we just put out a bunch of positive magnets and they'll all go to it? Nothing's that simple, right? But it does affect the cell and sometimes how it bonds or is attracted to things. So teichoic acid is in there. If it has a lipid moiety, it's still the same thing. It's a negative inclusion, negatively charged inclusion body. No, negatively charged inclusion molecule. Inclusion bodies, I'm sure, will be different. We'll hit those also. Okay, questions on the gram-positive. Pretty neat stuff, not too hard. You got it? Ready to move on? Okay. You got the signal, let's go. Okay, negative. You're going to recognize some things. Cell membrane, or cytoplasmic membrane, whatever you want to call it. Okay, now, peptidoglycan. It shows like it's pencil thin, yes. Many less layers. It's variable. Some have several more, but they do have a layer. A lot of people get mixed up on the cell wall, the cell membrane. What else do we have? Now we have an outer membrane. Okay, so in a way, there's two membranes, in a way. So the inside of that membrane is very much like the cytoplasmic membrane. The outside has a bunch of, it's a different molecule. It's a lipopolysaccharide, an LPS, and we'll talk about that. Now, between, and much more important, is what the different books call this different things. The periplasm is one thing. Sometimes they call it the periplasmic space, which I wish you wouldn't think about because it really isn't a space. That was an artifact of studying it. But if you want to call it the periplasmic matrix, that's fine. In a way, think of it as kind of cytoplasm. Okay, that's what's kind of inside there. Not as complex as that. What we find out is that that periplasmic matrix is really important for endocellular, so inside cell, endocellular enzymes. So it's an area of digestion and also a place where, of course, protons can be put without being lost to the solution, thus you can have proton motor force. Okay, so in the gram-negatives, we've talked about the cytoplasmic membrane. We have our peptidoglycan layer. What looks like a space, we've talked about that. And then we have the outer membrane. Now, the inside part of this outer membrane is still the little circles and two tails that we use down here. Is there a little bit of variation on the molecular level? Yes, it seems to be. It's nature, right? But by and large, this lipid layer is very much like this lipid layer, or this lipid layer turned upside down, the same thing. Okay. Now, this next part of the outer membrane, this is why it's not just called cytoplasmic membrane 2. It's a little bit different, right? This outer membrane then has these, you see it drawn here as hexagons, okay? These are polysaccharides with a linkage that's a small lipid. So you're like, oh man, I'm going to have to learn a bunch of stuff. No, we're just going to keep it kind of simple at the structural level, but realize that we have a bunch of sugar moieties coming out of here. We kind of have this lipid A core at that head because it's not exactly a phospholipid anymore. So let's take a closer look. Either this slide or the next slide should get us closer to it. Yeah, here it is. I thought so. So if we're to look at this, let's start with, so what's going on here? We got the cell membrane on the side. Here's the cell membrane, okay? Here's the periplasmic matrix. Here's the inside part of the lipid bilayer of the outer membrane, and here's the outside, okay? So we know how we flip this thing through space. So here's our fatty acids in there, and now we have our glucosamine sugars starting in here. KDO is deoxyketoic acid is a common part of the outer membrane, and I don't know why. I did some analysis on it, a postdoc, because I was trying to quantify this stuff. but it's a common moiety on that. And then this part can be variable. So here we have a core polysaccharide that's a little bit more conservative, doesn't show as much change. The O-specific polysaccharide can be quite variable, and thus you see the N. That means you could have many repeating, in this case, trisaccharide chain, or basically you can have heptasaccharides, hexasaccharides repeating in here. This is very variable. It can be fairly inert, this part, and just be really sticky, like cotton candy is sticky. Okay, it can be that kind of thing for the bacteria. In some cases, the O-specific polysaccharide can cause disease, like in salmonella. It'll interact with membranes in your body and make you very upset. So we'll keep an eye on that in chapters to come. But that, when they say, oh, the O-specific polysaccharide, you go, oh, oh, the O-specific, I know where that is. So that's the deal there. Let's just, before we leave the gram-negative, just a few things that, I don't know if the book covers it or it's in a lecture, but the thing that makes the gram-negative such a prolifically diverse organism on the face of the earth is kind of this, in some ways, the reason why coleopterans, why beetles are successful as a life form in the insect world. Okay, you know, it's a type of model of body type that really works well in a lot of different situations. For the beetles, you're a little bit hard, you're soft enough where you can move around. You know, you got some crazy looking antenna coming out of there. Sometimes, you know, lots of different things. You can resist desiccation, on and on. So beetles, you know, all over the place. Okay. It's true, there's a lot of species of archaea. There's a lot of species of gram-negative bacteria. The gram-negs do really, really well, though. The greatest diversity probably is in the gram-negs. And if you think about it, the cell membrane is pretty ubiquitous. It can't be there. It's that outer membrane. Let me go back a few slides. If you want to take a nap at this point, that's fine. I'm just going to give you a few of my thoughts. But this is not special to that cell type. What is special is this, okay, and what goes on here. This has a real sticky component to it. The thing that, before we leave, I should also tell you, is that some gram-negatives have other sugar strands, and those are really long, okay? They put together, that's called the glycocalyx, the sugar coating, right? The calyx, you know, holds up the flower. The calyx is an outside, and the sugar coating. The glycocalyx, and this you should probably write down, this does become important, right? The glycocalyx, which is external to this, not always present, but in a lot of them it is, the glycocalyx, if it's loosely meshed, okay, it's just really diffuse, they literally call it slime. There is actually a definition of slime. It's not just for the movies anymore, right? If it's compactly put together, then it's called capsule. Okay? That really, we think of a capsule perhaps as something that goes to the moon, or something we eat medicine with, or vitamins. We think of it as a real defined thing, but the capsule is more tightly knit sugar strands. Let me tell you about the capsule a little bit, and we'll get back to the adaptability of the gram-negative. What's cool about, as you know, if you had a big sponge made out of sugar, imagine if you had cotton candy at the fair, and instead of biting into it, and you get this sugar rush and your eyeballs pop out and all that kind of stuff. Instead of that, instead of having a bond that's broken down by water, that the very fine strands are insoluble in water. Just stick with me on that idea. Maybe you can think of a sponge or something, same kind of thing, but lots of fiber, okay? If you took it and stuck it in water, that cotton candy thing, if it didn't dissolve, would be like a big gelatinous mess, right? It'd be like big and slimy. It holds a lot of water. That's the same kind of thing. These sugar strands, they're not soluble, and they pick up a lot of water. So the capsule can be up to 95% hydrated. You go, so what? The so what lies in is that the capsule then acts like a little water reservoir when the cells encounter dehydration. Okay. It also changes their shape. They might not settle out of the solution as fast because you're changing its buoyancy. A little bit can go a long ways. Okay. Also to you, that is where the slimy feeling is in most cases when you pick up something that's been lying in the water for a while. Let's say you see a leaf. Oh, it's a pretty leaf. And you pick it up out of the water and you go, oh, it's slimy, it's gross. What's happened is a biofilm has gone across there, and that matrix, that polysaccharide matrix, is holding all the water, so you're slipping across the water just like you might on the sidewalk on a rainy day. It's a lubricant in that case. So it's neat from those standpoints. So the capsule in the gram-negative can help you stick, okay? Klebsiella pneumoniae sticks to your lung like no one's business, and just gets that big sugar thing, and just now you got to try to cough it out or whatever. Get antibiotics. You get the idea. Sticks. Okay. The capsule has a water reservoir in nature. It can help it on its buoyancy. So there's a lot of adaptation there that the gram-positives just don't have. They're little kind of rubbery balls when it comes down to the level of their scale. They have that peptide, but they're cool. Got a lot of them in your mouth right now, a lot of them in your nose, but they're different. Now if we move in, okay, the other thing with this area is we found out that this can cause disease to the host, okay, but it also is an area that can trap molecules. In nature, the capsule and the outer membrane, it's a problem. Again, diffusion is a problem. You kind of see a theme here. Maybe I should think about diffusion as an essay question. The diffusion is a problem because let's just say you want to degrade polysaccharides. You want to degrade oil molecules. If you're how do you get them into what is the limit of getting cells through the cell membrane? Yes, 600 Daltons, about the size of maybe around 6 cellulose molecules. You go, well, cellulose, glucose, that's all you need. Well, there's other stuff in the world. You don't always just have free glucose laying around, because if there's glucose in your body or in your water, every other microbe wants a hunk of that, right? I mean, it's in competition. So now you got to say, well, there's a nice piece of starch over there. I got to get at that and get that glucose into me. So you start spewing out enzymes. If they're external to the cell, they're called exoenzyme. Makes sense? Okay. If you're spewing out enzymes into the water, what happens? Diffusion. Okay, some hit the mark, some don't hit the mark, and you're spitting out and working day and night, trying to make energy to make more enzymes, and you're losing your energy, because you're trying to get the energy from the molecules you're trying to break down. So the gram-negatives say, well, it's enough of that business. I'm just going to stick onto that starch. I'm going to kind of get some of the subunits into here. Okay. Then this area, so exoenzymes, ectoenzymes in here, okay, not endo, I said that before, my mistake, ectoenzymes in here start to degrade things more, okay, and now you can bring it into there. The things are miraculous, they're everything from humic acids in soils to oils to fats to all kinds of things, okay. You don't see that kind of degradative power amongst the gram-positives as much. So, I'm going, why is he telling me all this? Back to our theme of diversity, of common themes. We have a the theme of diffusion we've talked about before with electrons and protons, with enzymes, and diversity due to simple morphology. Okay, so and you might have a page where you're keeping notes on that, and I'm kind of going, hint, there's an essay question coming. I'm trying to help you on this, but also that you can learn and start thinking about common themes as you go through complex subjects. Some of you are going, yeah, I got it. Okay, good. I'm here for you, man. I'm just trying to help out. Okay, now, the Gram stain. This is pretty neat. In the 1800s, Johannes Gram figures this out. What is neat about the Gram stain is it is one of those moments, it's eloquent, because it's simple chemistry coupled with dyes, coupled with cell morphology. And we use it to this day. Oh, we got all these molecules, we got all these computers, what the, No, I mean, one of the first things, they say, oh, do you have a gram-positive infection? Do you have a gram-negative? Because right away, that tells the doctors something. It tells people something about that cell. So what's up with the gram-positive, gram-negative? You guys did this in freshman biology? Anybody? Okay, tell me how it works. Go ahead. They tell you how it works? Do you want to know how it works?
It stains the layer of peptidoglycan.
Partially right. They told you. Actually, it's not right. But you're right in that it has to do with the peptidoglycan. That's what they tell you. Go ahead.
Okay, so they add like this purple dye, so it absorbs it, and then they rinse out that purple dye, and one of them will hold that dye, and the other one won't.
Okay, got the steps. Let's get down to what it works. And you're right, and you're partially right too. I don't like want you to cut my tires on my car or something. So okay, here's the deal. You're right on the steps. Now let's put some definition into it. We all kind of remember. Oh, it has something to do with colors and the cells, and that's great. Okay, let's get down to some science here. So we make a film of our bacteria. We grow up a pure culture right now to keep it simple, okay? And we heat fix that. The first thing we add is a purple color, or violet, crystal violet. Now you're right, the cell does stain purple. All the cells, any kind of cell, gram-positive or negatives will get purple at this point. It's really a basic dye, and it's going to the chromatin. To some degree, the peptidoglycan hangs on to it, but the important thing is that the peptidoglycan stops it from going out of the cell. This is where it gets clever. Okay, we're already done. So we're skipping a few steps, but we pour dye all over everything, and then we wash it off with water. Get rid of it. Everything's purple at this point. Yeah, okay, it's purple. So it's not at this point. It's not differential. It's like, oh, if we stop here, I got a bunch of purple cells. Got it. Let's move on. Now, the next part. This is crafty, and you left it out, but I know you guys did it. You did a good job remembering it. You add iodine. Why? Now maybe you're starting to remember the pieces. Why add iodine? I put iodine on my cuts. I put iodine I use it as a disinfectant, on the countertops. Why put iodine on a simple, now dead culture that I just flamed to a slide?
It doesn't react with peptidoglycan, at least in the gram-positive cells, but since they've got outer membrane layer and gram-negatives, they can't reach.
But they both have peptidoglycan. You're partially right. You're on the trail, and good for you. He said it bonds to the peptidoglycan. They both have peptidoglycan, so it's like, well, would I get like faint purple and dark purple? We really get purple and clear after this step. So partially there. Good thinking. The deal is iodine is called a mordant. 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 Okay. You go, so what the heck is a mordant? A mordant is a chemical oxidizer that complexes with a stain or a dye. Nothing you really probably learned about early in your childhood. You didn't watch cartoons going, oh, by the way, kids, here's a mordant. So this is probably a new word for most of us. Fine. Okay, but what it does then is it grabs onto the crystal violet and it complexes with it. So now it's harder to wash out. You can still wash it out if you have a leaky outer membrane and cell membrane, but you can't wash it out if you got many, many, many layers of peptidoglycan. A few it'll get through, but it won't get through all of them. This is the cleverness of the Gram stain. This is eloquent, is to think about how to take stain, because if you just try to put in a big stain to begin with, it's not going to go in right. The idea is kind of like to sand in the first part and then lock it in there. That's cool. We do that trick now to this day with different stains, with fluorometry and different things. It's right here. So now we put it in. What color are all the cells? They're still purple, and actually if you look at them, they're more purpley, the word. Okay, fine. But we're going to wash that off with a decolorizer. You may have used alcohol. People use acetone, a little bit less forgiving. I let students use alcohol yet, but the med techs use alcohol, acetone alcohol, that's fine. Basically the idea though is to take that stain and wash it out. Okay, now here's the rub. The complex stain is trying to get out, it's getting pulled out, by diffusion, and it can't in the Gram positives. Too many layers. The Gram negative has a cytoplasmic membrane, it gets through that. It kind of gets caught up a little bit in the peptidoglycan, but there's so little layers of it, it still is being able to be moved out. The outer membrane is not a real problem, that's real leaky, a bunch of lipids and stuff. Actually, to an extent, the alcohol acetone will dissolve some of the lipids, actually, to some degree. So that's another trick they sometimes leave out, but the main idea is still here. Now, at this point, so at this point, if I have a Gram positive cell in that initial smear, what color is it? I try to wash out I hit everything crystal violet, I try to wash it out. Gram positive, is it purple or is it what? Yeah, okay, can't get it with you. The Gram negatives, what color are they? They're colorless. Colorless cells are hard to see. One of the primary reasons to use stains is to increase contrast. We know that. We even have a contrast button on our iPhones. Okay, so the thing is to really make it stand out from the background. And these things are small, they're clear. My gosh, if you don't have your microscope set up exactly right in color, no hope of seeing them. So they said, well, let's take the guess out of this. Let's get this going. And so they come with a counter stain. A counter stain of red dye called safranin, S-A-F-R-I-N-E-N. I don't know if it's up there, but that's what it is. Red dye is fine for this class. You'll get it again in lab if you're in there. Either way, now my Gram negs are red, or sometimes people call them pink. I like to just say red. I'll tell you in a minute. And the Gram positives are violet. All right. Now in the beginning, I think everybody was kind of going like, yeah, one of them turns purple, one turns red, and it's real easy to mix them up. Everybody does. I mix them up. So I'll just tell you what I use, because I'm not a very complex person. I say positive purple, okay, PP, and then I don't try to go, don't say pink, because then now you're mixed up again. Positive purple, Gram positives are purple. Okay, oh, that's pretty simple. Yeah, but if you get mixed up, unmix yourself any way you know how. So that's that. Isn't that cool? Dissolving a little bit of the lipid, okay, complexing that first stain. These are basic stains. Basic stains go to DNA, and using a mordant to complex it. Very, very clever, and relating it to the cell morphology. When you have a Gram positive infection in your throat, people immediately say, well, are you allergic to penicillin? Because they're thinking about that kind of antidote for your malaise. All right. gram-negative, well, there's different tricks, depends on where it is, and now we don't wanna knock out all of your flora, we try to be a little bit more specific, you know, cause there's other problems in knocking out stuff throughout your body. Do you get the gram stain? You good with it? You could explain it? You excited about it? Go home, tell your folks, tell your boyfriend. Oh, you'll never guess. The archaea are different a little bit. We're going to talk about this. Unlike the ether bond that we can say we take to the bank, archaea have that ether linkage. That's when we can look across the domain and say. What I'm going to tell you here are some things that are in some of the groups and not the others. So in a way, these are general characteristics, but they're not universal characteristics. On the other hand, we're learning about a new domain. Let's go for it. Okay, N-acetylglucosamine still has it in the cell wall. Okay, but instead of muramic acid, it puts in this Toloso group in there, so you kind of have this a different side chain on there. The N-acetyl part's the same. It's the Toloso amino uronic acid that makes it different. The bond between the different saccharides is not a 1,4, it goes 1,3. Why? I don't know, but it is a little bit more stable bond in some cases, and it might help in some of the adaptations it does. But when you turn it from a 1,4 to a 1,3, you know, that kind of thing, you can hit the archaea with lysozyme, they won't degrade. Some people do that. They'll get a culture, they're trying to typify it. Is it sensitive to lysozyme or muramidase? So there's that thing. But other than that, those parts are pretty much the same. Together, they call this not just muramic acid, but pseudomurine or pseudomuramic acid on that. Some archaea have a layer of protein on the cell wall. The type of configuration, it's called, is an S layer. Okay, consists of protein or glycoprotein. And this, of course, putting a protein shell around a sugar shell makes it, or a lipid shell, makes it very resistant. So some of them have an S protein layer. Well, I know. Why did I put the mycoplasma in there? Because we talked about mycoplasma on lectures. So mycoplasma are gram-positive. It's kind of strange. You don't really have a cell wall, so wait a minute, how do they gram stain them? It doesn't mean anything in that group. This stuff doesn't work everywhere. But they know from the DNA that belongs in that group. But with some of the archaea, some just lack a cell wall. Okay, this guy, thermoplasma, is small. These things live in like really hot places, as the name implies, plasma, in that they don't really have a good cell wall. You wonder how they do it. Their cell membrane is wired differently. But yeah, so. We see this characteristic amongst the bacteria, and a few of the archaea have that also. So as a good characteristic, you really couldn't take that one to the bank, but it does exist. From this point in the lecture, I just broke it up into 3 conceptual parts. You know, we talked about the membrane, we talked about transport, took that apart, then talked about cells. Then we went into the cell wall. I like to emphasize it because it's really, really important. What we want to do in the last part of the lecture is move cells around by locomotion. I don't know if I'll get to it today, but I put up some really nice little videos for you. Some videos are just eukaryotic motion, and I make the distinction because eukaryotic flagella are different from the bacterial flagella. We'll talk about that. So I did put up a video on how the bacterial flagella are put together. They work like little propellers. So we're going to talk about moving. Oh, and I put up a slide or a video that a graduate student and I did, Mandy Cook. I love working with Mandy. We would take little pipettes like you guys use, and we would heat them and draw them out into capillaries, and then she would dip them in agar that had different things in them. She was testing for chemotaxis. Yeah, I point to my nose as though bacteria have noses, right? Well, anyway, chemotaxis. So she'd have, the one I'm showing you is there's some phosphorus in the agar, and there's a group of pseudomonids that we knew had chemotaxis. So they're swimming around in this well of media, and they get it, and within each frame is like a portion of a second. And you just see these guys just swarm to it. And you get the idea, these things move really, really fast. So those videos are for you, whether I show them in class and I watch you watch them, or you watch them at home. Take a look at them, they're a lot of fun. And then we'll talk about inclusion bodies, and we'll work our way around the other parts of the cell is the thing. So, oh yeah, I thought I put that in there. Okay, what I thought we could do is like a little memory check. Hey, I'm really happy you showed up the day before, the day before you have a short break. But also you showed up, I thought I'd give some extra credit points. You're not going to get rich on these, but they don't go, they're not divided by the quotient, the divisor in your average, so they're on top of, we're on top of things. So I got 5 little questions. Let's take that. Well, what I'd like you to do, these are index cards. On one side, print your name. I can't give you points. Okay. And you just number 1 through 5 on it. I have letters. Just print the letters. I don't think this will take you that long. On the back side, I usually ask people for introductory information. Now, I don't want personal stuff. No, I don't. I mean, I like you guys, but I don't want anything but I would like to kind of know what you're doing in biology. I mean, just something general. I want to be a doctor, or I don't know yet, or just tell me why you're here. I know why you're taking the course. You're a major. They're making you. Otherwise, the room would be empty. I get that. But on the other hand, just tell me that part about your professional self, where you're going in life.